Well, good afternoon, everyone. I want to thank you for the invitation to be here today, especially because of the opportunity to share the work that I am doing in the group to which I belong, here in Spain and in Argentina. I will talk about polymeric particulated carriers in drug delivery, especially about its obtention, study, and characterization. As I said, I am here as a PhD student visitant. I just want to tell you a little bit about this. I am working in the Institute of Polymer Science and Technology here in Madrid. And in Argentina, I belong to the Polymeric Matrix Composite Materials group. Well, I will continue with my talk. And yeah, one important thing, because now I will present some of her results. Here in Madrid, I am with the PhD Nicoleta Resignano. And well, I will give a little outline just to organize a little bit the presentation. Of course, I will talk a little bit about drug delivery devices, but I am sure you are familiar with those concepts. And then I will talk about the two biopolymers that we use to make our drug delivery systems. And finally, I will talk about three systems that we have studied in both groups. And I have to say that some of the results are preliminary, but we have trust that they can continue growing. Well, as I said, I am sure we are familiar with the definition of a drug delivery system. The important thing that I wanted to say here in this part is that besides all of these general objectives that we are chasing all the time in this field, we have to always take into account our couple between our vehicle and our active ingredient. In this case, I will talk about polymeric micro and nanoparticles, and that's why I was talking about that. We have to choose our polymers, and we have to know how they behave with our specific active. In this part, I will only give a little bit of information of the different kind of polymeric nanoparticles or microparticles, because we can get both. And the main thing is that although we can have nano or micro capsules in which we have a core shell structure, in which ideally we have the drug in the cavity of the core, we can also have matrix nanoparticles, and these are the ones that we work on. In that case, in matrix nanoparticles, polymeric nanoparticles, we try to achieve that our drug could be trapped into or within the chains of our polymers. And I also put the other example because I have seen some of this in literature in which they use the matrix particle only to attach the drug on the surface. As you can see there, we can encapsulate or trapped or covalently attach our active ingredients in order to get our biotechnological goals. So, as I'm sure you also know, Plga has been one of the most successful biopolymer used. It's approved by the FDA. In our case, we are going to use Plga, and we use it because of the degradation time that Plga exhibits. In this case, as you can see in the chart, we have a 3 to 6 months of the degradation time, but you should take into account that we are talking in the chart about the pellets of the Plga. So we can say that if we have a nanoparticulated system, this time of degradations will change in order to achieve practical degradation times for drug delivery, for modulate the release of our active, or the goal that we wanted to reach. Also, in one of the systems that I'm going to present here, we use PLLA. And in this part, I only want to say that besides that the polylactic acid has a higher degradation time, as you can see there, you have from 12 to 18 months. What the strategy that we try to use is to get copolymers between polylactic acid and polyethylene glycol in order to can get a better degradation time in our systems, and also because of the requirements that we must take into account when we are talking about intravenous administration. In that case, get a copolymer between polylactic acid and polyethylene glycol is very useful because of the functionalization of the surface. Well, the first system that I want to talk about is a previous work that we have made in the group here in Spain, in which we could get bobine serum albumin loaded Plga nanoparticles. Well, this is a pretty challenging thing because of the work that you should take on mind when you are talking about hydrophilic drugs. It's always a little bit more challenging than an hydrophobic drugs, and in this case we used a laboratory method. It's the double emulsion technique, and well, as you can see, this method requires you to make 2 emulsions in order to get an aqueous suspension of nanoparticles. And in our case, we lyophilize or we freeze-dried that aqueous suspension of nanoparticles in order to get our system. Then the system could be resuspended and use it. So we are always talking in this presentation. We will see double emulsion technique with solvent evaporation, and we also are going to see simple emulsion technique when we will talk about an hydrophobic drug. In this case, with this technique, we could obtain the nanoparticles, and the size, as you can see, was 283 nanometers, and the polydispersity was really good. And the encapsulation efficiency in this case, it's really good in considering that it is a double emulsion technique. In a double emulsion technique, you can have from 30% to 60%, but it's not common to have higher encapsulation efficiency. The interesting thing is that these nanoparticles were tested in stem cells to check their internalization. And there's a figure that I didn't include in the presentation because it would be so long, about the measurements that we did after 3 hours and 6 hours of started the test. And on that point, you cannot see fully internalization, but from 12 hours, you can see how the stem cells appears in all the images. I mean, all the stem cells were positive from 12 hours and beyond. And these are a really promising test that we have done. Another thing that it was very interesting is that when we finished that test after 72 hours, that was the maximum time, the stem cells were continue being monitorizing. And what we can see is that the fluorescence started to decrease little by little. And this was interesting because this shows us how the polymer started being degraded. And once the polymer starts the degradation process, we can also could check that the bobine serum albumin that was bonded with FITC started also to degrade from the FITC. So we start seeing that the fluorescence start to decrease little by little. But this means that the nanoparticles, the polymeric nanoparticles, started to release all the bobine serum albumin that was trapped there. Another really interesting test that we performed, it was an in vitro degradation of the polymer. Of course, that we have different phenomena that appears when we are talking about the release of a drug from the polymeric matrix, but it was very, very useful for us to see what was happening with the polymer. So we observed some morphological changes. Once we take our samples from 7 14 and 21 days of incubation with PBS and at 37 degrees. What we can see is that in day 7, we can see that the particles are still spherical, which is the common morphological feature that we always observed. But we start seeing like pores in the surface of the particles. Then at 14 days, we see how we start losing the spherical shape. And then on 21, and on day 21, we see that we have no longer any nanostructured feature, and how the polymer starts to get aggregated without any pattern. And with PLA that we also tested, we can see the difference. That takes a little bit longer to see all these changes. Well, regarding this protein, as I said, as we tested BSA, we decided to continue testing some hydrophilic agents. And in this case, we started to work with polylysin loaded PLA, PEG, PLA microparticles. In this case, we used a blend of polymers, PLA alone, and then the copolymer with PEG, as I said. And the objective was to study this kind of peptide. This was a polylysin of 10 units, because in our group we are studying a collaboration that involves peptides for anti-cancer treatment. As these peptides are really expensive, we decided to use polylysin as a model. So we get some polylysin microparticles, but we did it with a different technique, not an emulsion with solvent evaporation technique, but in this case, as it was a collaboration, we use a proprietary electrohydrodynamic technology that allow us to get the particles in a more versatile way and with a really high efficiency considering an hydrophilic drug. We get these particles. The shape is different from the ones that we saw before because of the technique that we used to obtain it. That's why they are all a little bit bigger than the others because of the shape that they exhibit, in order to have the suitable quantity of polymeric matrix to release the drug. As you can see, in this case, we only use a ratio from polylysin to the polymer of 1%, and the encapsulation efficiency, it is really good for an hydrophilic drug. It was 81%, and well, the size, as I said, it was around 1 micron. Regarding these particles, these microparticles, we checked that the thermal stability of the particles was okay, and we can also observe that there was an association between the polymer, the blend of polymers and the polylysin. With confocal microscopy, we also checked the presence of the polylysin in the particles as a previous study, because the polylysin was also attached to a fluorescence unit. As future work regarding this specific system, the polylysin system, involves a release profile, because we have made a release study using a France cell, but now we have to quantify all the samples that there are around 300 to check how is the release of the polylysin from this matrix particle, because in particular these peptides that I told you about are considered for a weekly treatment for a specific type of cancers. So our idea is to check if we can get this kind of release profile for this kind of application. And also, of course, as I told you, these were preliminary results. We want to check cytotoxicity and internalization, because another interesting thing regarding the other peptides is that opposite to the common thing that we used to chase in nanomedicine, they don't need to internalize into the particles. If they internalize into the cells, sorry, if they internalize, we lose the therapeutic effect. So, as I put there, the idea is to extrapolate all this to these innovative oncologic peptides. Finally, we also work with an oncologic agent, tamoxifen, and the interesting thing is that we could study a different drug regarding the hydrosolubility. It's an hydrophobic drug, so in this case we use a simple emulsion with the solvent evaporation technique, so we only have to make one emulsion. We use always to make the emulsions tonications, and we use as a surfactant PVA. Another interesting thing is that in this case we try to compare different surfactants, and we try to use also poloxamer, but in this case it was not good for us, so we decided to continue with the PVA, with the polyvinyl alcohol, and with this surfactant we can get really monodisperse particles. We get with a 10% of tamoxifen in relation in relationship with the mass of the polymer, we get particles around 200 nanometers, and with a really good polydispersity. The efficiency and the encapsulation efficiency could be improved because we started not so long time ago with the synthesized, so we will try to optimize it because we know that we can reach 80% in encapsulation efficiency. Another interesting thing that we did is to get our freeze-dried particles and resuspended it and tested versus temperature to see how these particles behave when we tested with temperature regarding specifically the temperature that we can find in the biological environment. And we see that although the size changed a little bit, this difference in size does not reach 10%. So for our resuspension, it's pretty good. And the polydispersity, it's also good because we don't have a really high variation. And as you can see in the scanning electron microscope images, also the shape, it's always spherical, and we can also confirm the narrow size distribution. With these particles, we did a collaboration with the biomaterials group in the Institute of Science and Technology of Polymers here in Madrid. And what we saw is that when we tested the particles, the Plga particles loaded with tamoxifen in human umbilical vein endothelial cells, we see that when we compare the empty nanoparticles, Plg nanoparticles, with tamoxifen-loaded nanoparticles, in the two more concentrated suspensions of nanoparticles, we see a statistical significant difference. So we see that in these two concentrations, the nanoparticles loaded with tamoxifen are in fact toxic. And what we wanted to do later on, the next step for us is to test these same systems, but using a breast cancer cell line in order to check for selectivity and cytotoxicity of tamoxifen, but with the cancer cell that is our target. So that's the next step in regarding in vitro test. And also we will test release profile, and we are trying to do some kind of innovation in the route of administration of tamoxifen, because as you may know, tamoxifen, it was deeply studied regarding oral administration, but as oral administration has several side effects, recently in literature they have started to appear some topical and transdermal administration, and we would like to try if we can administer tamoxifen with in this route. And that's why we are also going to, we started last week to make some nanofibers of PLA loaded with tamoxifen also. And the other strategy that we wanted to try is to use some copolymers, but with the opportunity of attach some specific ligands in the surface to do active targeting. I have to thank all my group from Argentina, as well as the new group and new friends that I have here in science. And I also wanted to show you a picture of my city, because you are always welcome. And I thank you for hearing me today. Thank you very much.
Questions for our speaker? No questions? Okay, thank you very much once again.
Thank you.
That's your certificate.
Yeah.